Chapter 5, the erythrocyte or red blood cell. Erythrocytes or red blood cells carry oxygen from the lungs to the tissues or the tissues of the body cells. Here, it delivers oxygen and picks up carbon dioxide, where it is a waste product, and delivers it to the lungs to be exhaled. If there is an insufficient number of red cells, characterized by an inadequate tissue oxygenation, this term is called anemia. Now we're going to talk about erythropoiesis, or the making of a red cell in maturation. Normally, a mature red blood cell has a lifespan in the circulation of about 120 days. When we discuss the erythron, this is the total stages of erythrocyte development. This occurs in the bone marrow as a precursor cell, and as it matures, it gets smaller and the nucleus is extruded. Then it is sent into the peripheral blood, where you see a nice 7 to 8 micron cell that has 1/3 of central pallor and having a pinkish red composition. Erythropoiesis happens in an orderly fashion. It requires the major hormone, erythropoietin, in order to have a production. After its lifespan of about 120 days, these cells are aged and are sent to the liver to be destroyed. They are destroyed by either the liver, spleen, bone marrow, or the macrophages within the cell. After a stem cell has committed to becoming an erythrocyte, a burst-forming unit specific to the erythrocytes forms many colonies in vitro that takes about 10 to 14 days. In this stage, there are several hundred or thousand hemoglobin-containing red cells that are made as precursor cells. This gives rise to the next stage of the colony-forming unit, erythrocytes, which also has a very high potential to make more cells. The colony-forming unit for erythrocytes give rise to a discrete number of hemoglobin-containing cells that takes about 2 to 5 days. They have a very high concentration of erythropoietin receptors, and this is the immediate precursor to the maturing stages of the first identifiable red cell precursor called a pro-normoblast. As erythrocytes mature, they have a series of maturation stages they encompass. They have been identified in 6 different classifications: pro-normoblast, basophilic normoblast, polychromatic normoblast, orthochromatic normoblast, polychromatic erythrocyte, and the erythrocyte. How I remember this acronym is by saying PB Pope or Peanut Butter Pope, and I have remembered this acronym for many years. So let's look at the first maturing stage of a pronormoblast. The arrow is pointing to a very immature red cell precursor. It is the earliest one and produces 8 to 32 mature red cells through 3 to 5 cell divisions. It is the largest of the series and about 20 to 25 microns in diameter. It has a very high nucleus to cytoplasm ratio. The cytoplasm contains large numbers of ribosomes, and here is where protein assembly occurs. It stains very deeply basophilic or purple. There may be a pale area within the cytoplasm, which represents the Golgi apparatus. It's pale because it does not take up any of the stain used in hematology. There are very small amounts of hemoglobin present here, but they're not visible by light microscopy. The nucleus is very large and takes up about 80% of the cell or more and stains bluish purple. The chromatin is very fine and described as lacy. It has a coarser pattern than a white cell blast, and that's how you can kind of identify it, the differences. And it may contain 1 to 3 faint nucleoli. The next stage is a basophilic normoblast. It looks similar to the pronormoblast, but it's smaller. It's about 16 to 18 microns in diameter, and it is actively dividing. The cytoplasm is also deeply basophilic due to the increasing number of ribosomes. Here you will see that perinuclear halo around the nucleus, which corresponds to the mitochondria, which also does not take up the stain. The later stages have more hemoglobin, which causes the cell to have a lighter blue hue and start to have a pinkish hue. The nucleus to cytoplasm ratio is decreasing slightly, where the nucleus now occupies about 75% of the cell. The chromatin is coarser here than the pronormoblast. You see a dark violet hemachromatin interspersed with the lighter staining euchromatin, which looks like a spoke of a wheel. Here you may or may not see any nucleoli. Now let's look at the next stage, the polychromatic normoblast. Here the cells are about 12 to 15 microns in diameter. The cytoplasm is present and now is taking up a bluish-gray appearance, and this is due to the synthesis of large amounts of hemoglobin now and the decreasing amounts of ribosomes. The nucleus here, the nucleus to cytoplasm ratio is decreasing due to the condensation of the nuclear chromatin. The chromatin is irregularly and coarsely clumped due to the increased aggregation of heterochromatin. This is the last stage capable of mitosis. The orthochromic normoblast is about 10 to 15 microns in diameter and has a very low nucleus to cytoplasm ratio. The cytoplasm is now predominantly pink or salmon in color due to the concentration of hemoglobin. Some of them may retain a very small tinge of blue due to the remaining ribosomes in the cytoplasm. The nucleus now is very heavily clumped. It is the last stage where the cell has a nucleus. It is starting to become extruded while the cell is still in the island, or as it's lost as it's passing into the peripheral blood. The macrophages surrounding it will engulf the red cell's nucleus and dispose of it. The polychromatic erythrocyte, or also known as reticulocyte, has an irregular, lobulated, or puckered shape following the extrusion of the nucleus. Here the cell is remodeled. It's eliminating the excess membrane and it gradually acquires that biconcave shape. It still may have a bluish tinge due to the residual RNA of the ribosomes and the mitochondria. This is called a polychromatophilic red cell or a diffusely basophilic red cell on the right stain. The remaining 20% of hemoglobin is made during this stage. The cell is about 7 to 10 microns. It's slightly larger than the mature red cell. The polychromatic erythrocyte, or also known as reticulocytes, are identified in vitro only with a supravital stain. You cannot see this cell on a peripheral blood smear. Using a new methylene blue or brilliant crescent blue, this causes the precipitation of RNA in the mitochondria, which appears like these purplish blue inclusions inside of the red cells. The normal percent of these reticulocytes in the peripheral blood is 0.5 to 2.5%. They contain a small amount of iron, which can also be stained by a separate Prussian blue stain. The spleen's job is to remove any of those iron granules that are present. Here are images of normal erythrocytes or red blood cells. They are normally 7 to 8 microns in diameter and compose a volume of 80 to 100 femtoliters. They stain pinkish to orange due to the acidophilic hemoglobin within its cytoplasm. It lacks any cellular organelles such as ribosomes and mitochondria, and any enzymes required to synthesize any new proteins or lipids. Now within the bone marrow, you have several niches where different cells aggregate to be made. They're called erythroblastic islands for red cells. They adhere to a macrophage, and a macrophage is essentially a monocyte inside of tissue. So the central macrophage is surrounded by erythroblasts and reticulocytes. This macrophage is called a nurse cell because it sends out cytoplasmic processes that maintain direct contact with each of those erythroblasts. Now the central macrophage will also phagocytize or engulf that nucleus or destroy it if there's any defective erythroblasts. As the cells mature, it loses the adhesion here and detaches from that macrophage so it can pass through a pore of the cytoplasm of the endothelial lining of the bone marrow sinus where it enters into the peripheral blood circulation. The erythrocyte membrane is very complex and essential for the development and function of the red cell. The erythroblast has many receptors for EPO, which is erythropoietin, as well as transferrin, because iron is needed to create hemoglobin molecules. The membrane's specific function is to selectively sequester any vital component and also allow any escape of metabolic waste products that have occurred from the assembly of proteins within its structure. The cytoskeleton provides red cells with strength and flexibility that needs to survive the very harsh environment of the circulation. It also regulates metabolism by reversibly binding and inactivating any glycolytic enzymes. It also balances exchange of bicarbonate and chloride atoms, which aid in the transfer of carbon dioxide from the tissues to the lungs and balances any cation and water concentration to maintain the red cell ionic composition. The composition of the red cell membrane composes of phospholipid bilayer protein complex, which has about 52% protein, 40% lipid, and 8% carbohydrates. The purpose here is to control the functions of transport, flexibility, and also provides the strength needed to survive circulation. This membrane also determines the antigenic properties of the red cell. The lipid composition here, about 95% of the lipid content of the membrane consists of equal amounts of anesterified, cholesterol, and phospholipids. The remaining lipids are free fatty acids and glycolipids. The glycolipids are located in the external half of the bilayer. The carbohydrate portions extend into the plasma, and it's responsible for the antigenic properties of the red cell membrane, which determines our blood groups. The protein composition of the red cell membrane includes peripheral proteins that have enzymes and structural proteins. The structural proteins are used to organize a two-dimensional lattice network. This is very important because it provides the skeleton of the red cell membrane, which supports the membrane lipid bilayer. It gives it some viscoelasticity properties that contribute to the cell shape, its ability to deform in smaller venules, and stability. Also very influential in the red cell cytoskeleton membrane. About 80% of calcium is found in association with its membrane. Elevated calcium can induce membrane protein cross-linking and acts as a fixative to stabilize the shape of the red cell and reduce any deformability when it goes through smaller venules. Its membrane and skeleton is responsible for the structural integrity and deformability of the cell. So a 7-micron red cell must be flexible enough to squeeze through very tiny capillary openings and strong enough to withstand the turbulent circulation. Its deformability is due to its biconcave shape, its viscosity of its internal contents, and the properties of the erythrocyte membrane help in this capability. Red cells really have an elastic extension capability. They resume normal shape after being distorted. Now, if there's very large or prolonged forces that cause the cytoskeleton to reorganize, this can lead to permanent deformation, and we will learn about abnormal red cell structures in the next few weeks. As well, if there's excessive force, this can lead to fragmentation. Now, in order for the red cell to be able to accomplish all of its functions, energy is required. It's required to maintain the cation pump, to keep hemoglobin iron in the reduced state, to reduce any sulfhydryl groups in hemoglobin and other proteins, and also to maintain the membrane's integrity and deformability. Now, let's talk about the metabolic pathways of the erythrocyte. The biggest and most common use of energy is the glycolytic pathway. Here, red cell obtains energy in the form of ATP from the breakdown of glucose. About 90 to 95% of the red cell glucose is metabolized by the glycolytic pathway or the Embden-Meyerhof pathway. Normally, red cells do not store glycogen. This depends on the plasma glucose for glycolysis. So, obtaining energy in the form of ATP from the breakdown of glucose glucose needs to be metabolized to lactate or pyruvate. Lactate or pyruvate is transported from the cell to the plasma and metabolized elsewhere in the body. The pathway's role is to maintain flexibility, erythrocyte shape, membrane integrity, and normal levels of cations. The hexose monophosphate shunt metabolizes 5 to 10% of the cellular glucose. It generates NADPH instead of ATP, where glucose 6-phosphate is oxidized in the first step by glucose 6-phosphate dehydrogenase. Here, NADP is reduced to NADPH. NADPH converts oxidized glutathione back to reduced glutathione. It's very necessary here because it maintains hemoglobin in the reduced functional state. Keeping hemoglobin in the reduced functional state also prevents it from any oxidative injury by reducing reactive oxygen species produced during oxygen transport, or certain drugs or chemicals. It also keeps it in the I2+ state. Hemoglobin with iron in the 3+ state or ferric state is called methemoglobin. This is generally dissociated from heme iron and cannot bind oxygen. So in the methemoglobin reductase pathway, it's specifically working on keeping iron within hemoglobin in the ferrous state. If it doesn't, oxygen cannot bind to the hemoglobin molecule. In the absence of this pathway, 2% of methemoglobin will build up and severely limit the oxygen carrying capability of the blood. The Rappaport-Neuerberg shunt pathway bypasses the formation of 3-phosphoglycerate and ATP from 1,3-biphosphoglycerate. The role is in regulating oxygen delivery to tissues. So it sacrifices one of the 2 ATP molecules produced during glycolysis, so oxygen is available to be released to the tissues. The regulation of production is very important because it depends on the rate in the bone marrow, and it also depends on the release rate from the bone marrow. The rate of red cell release to the circulation is balanced to the rate of its destruction. So an increase in EPO release occurs because of an impaired oxygen delivery to the tissues, or low intracellular oxygen tension. So EPO is very thermal stable. It's produced in the liver, and it severely increases when hemoglobin levels drop. Aging is a characteristic of the red cell, so erythrocyte destruction can occur by 2 different methods. Here, the loss of ATP and the loss of reducing substances results in the oxidation of the membrane proteins, lipids, and methemoglobins. Cells are unable to maintain the cell shape and deformability. They lose their membrane integrity. This results in the removal of the red cell. The red cell can be destroyed in 2 different methods. Extravascularly, so outside of the vasculature, which is removed by the spleen, bone marrow, and liver. 90% of old red cells are destroyed this way. They have a very efficient method of removal and conserving and recycling any amino acids and iron that are essential to make new red blood cells. It can also be destroyed intravascularly due to any red cell trauma or damage to the membrane. This will result in cell lysis and the release of hemoglobin directly into the vasculature.